Yeah, I don't like to use a pointer because,
Not like that because you have to turn.
yes, but also people just see a waving red dot, whereas it's better if you just describe it.
I use the mouse.
Yes, yes, you can do that. Because also online they won't see a pointer. Yeah. Yes. Yeah.
Pictures, pasting, stuff.
Yes.
Just
I have some.
point.
Yeah.
Blank ಆಗಿರುತ್ತಲ್ಲ. So we need to project itself slides. Copy item, huh?
That is not really getting in there. Share on WhatsApp and everything. Which number?
ಎರಡ್ ಸಾಕು ಎಷ್ಟೇನು ಮಾತಾಡೋದು ಅಲ್ಲಿಂದ ನಾವು
Like this.
Then but I am there for. Yeah, see, it not be Zoom link share.
Hello.
No, already it's open. Zoom link. Yeah, okay. Gourie-Devi ma'am is right. Who's Lakshmi? Lakshmi from physiology. Why can't they come? She could have come as Falda should have come from Kola. Can you ask MGD whether she has Zoom ಅಲ್ಲಿ ಇದ್ದಾರಂತೆ. ಎಂಡಲ್ಲಿ ಮೇಡಮ್ ಮಾತಾಡ್ಬೇಕು ಬಿಫೋರ್ ವಾಟ್ ಆಫ್ ಟೈಮ್ ಮೇಡಮ್ ಟು ಟೆಲ್ ಪೀಪಲ್ಸ್ ವಾಟ್ ಆಫ್ ಟೈಮ್ ಮುಂಚೆ
Hello. Hello. Hello.
A very good afternoon to one and all. We have all gathered here today for the prestigious third Dr. M. Gourie-Devi Oration, organized by the Department of Neurology and Institute as a token of our love and appreciation of the contributions of Professor M. Gourie-Devi, former director and pioneering neuromuscular specialist of NIMHANS. I am Dr. Seena, additional professor of neurology, and I am privileged to be MC for this program. Professor Gourie-Devi ma'am has contributed immensely for the care of patients with neuromuscular disorders. She was an excellent clinician and researcher and teacher and a world-renowned neuromuscular specialist who continues to inspire the younger generation with her pursuit of academic excellence. So this oration is an opportunity for us to express our love and gratitude to her for her immense contributions. Madam has joined online for this oration. And we have with us various eminent neurologists and neuromuscular specialists from all over the country. We have with us former director and retired senior professor of neurology, Professor P. Satish Chandra sir. We have former director, Professor Uma Maheshwar Rao sir. The former registrar, Professor Sankar Narayan Rao sir. We have the deans and associate deans, head of department of neurology, Professor Madranath sir, heads of various other departments, other faculty and colleagues from neurology as well as various other departments, residents and PhD scholars and other researchers from the departments. And we are also privileged to have neurologists from all over the globe. And we have online with us Professor Venkamma ma'am, a senior neurologist, and also Professor Anthony Urtis-Beria, a leading neuromuscular specialist from France. And we also have researchers from the UK Motor Neuron Disease Research Institute who have joined us online. A warm welcome to all of you. And we have with us an eminent orator, a leading researcher in neuromuscular disorders, motor neuron disease, Professor Amar Al-Salabi. The therapeutic landscape of ALS is evolving, and the clinicians, scientists, and patients across the globe are putting tremendous efforts to unravel the mystery of ALS and to confront this formidable disease. Professor Amar Al-Chalabi, with his vast experience on research and care of ALS patients, is going to deliver this oration on the mystery of ALS. Can it be solved? So at the outset, let me invite the dignitaries to come to the dais. Let me invite first the head of the Department of Neurology, Professor P. Madranath sir. I request you, sir, to please come. I request Professor Nalini Madam, Senior Professor of Neurology and Dean Neurosciences and leading neuromuscular specialist in NIMHANS, to kindly come to the dais and be seated. I request our orator, Professor Amar Al-Chalabi, Professor of Neurology and Complex Disease Genetics, Co-Director of UK Motor Neuron Disease Research Institute, London, to please come to the dais. I request Professor Satish Chandra sir, the former Director and Senior Neurologist, retired Senior Professor of Neurology from NIMHANS, to please come to the stage. I also request our registrar, I think the registrar is, yeah, I think he was supposed to come. I think he had some other pressing commitments, and our director ma'am also couldn't join due to some pressing commitments. Now, let me invite Dr. Satish Chandra sir to give an introduction about Gourie-Devi ma'am. It's my distinct honor and privilege to welcome him. He was the former director of NIMHANS, and he's a leading neurologist in epilepsy, demyelination, neuroinfection, and various other neurological disorders. He has worked with ma'am as a student and later as a faculty in the Department of Neurology. And it's very apt that sir is going to give an introduction about Professor Gourie-Devi ma'am. And sir has a few slides also and numerous memories to share with us. I welcome you, sir, to kindly give the introduction about Professor Gourie-Devi ma'am.
Good afternoon, Professor Nalini, Professor of Neurology, Professor Madhuranath, Head of the Department of Neurology, and the guest orator today, Dr. Amar Al-Chalabi, and the guests who are all come here at the invitation of the Department of Neurology, the staff members and the students here, and also my teacher, Professor Gourie-Devi, who is on the online, and also many of my colleagues are all there on the online. It is a wonderful day in the evening. We are now having a third Dr. Gourie-Devi's oration. at the outset. Right, can you switch it off a little bit? My pranams to my teacher, Professor M. Gourie-Devi, who was a teacher and a mentor for me. And Professor Gourie-Devi has seen generation and generation of students here. I think if I'm correct, there is a fifth generation of students here now in the audience of Professor Gourie-Devi. And you can wonder how young she is. She is, with the permission of the madam, she is just 88-year-old and very well versed with the computers and sitting on the computer with an online operating Zoom and watching this event. Can you imagine that? And you can hats off to her. And she is very, very attentive even today and joins for most of the meetings online. And that is what our professor and our teacher is there. So welcome, madam, for this oration, which is a prestigious oration for all of us. Next one doesn't move. Yeah. Professor M. Gourie-Devi is currently working as an advisor in the Department of Neurophysiology and senior consultant neurologist, Sir Gangaram Hospital, New Delhi. And she is now Emeritus Professor of Neurology, Institute of Human Behavior and Allied Sciences, New Delhi. That is what I wanted to emphasize. Now she is continuing to be Emeritus Professor and going to these institutions once a week and even teaching now and participating in the projects. She has a number of projects even today to work with various younger colleagues and inspiring them. Then honorary advisor in the neurological research in the Indian Council of Medical Research. And she was the professor and head of the Department of Neurology at our institute, the National Institute of Mental Health and Neurosciences, from 1997 to 1977 to 1997, 20 years, 2 decades she was here. And when I was a student here, and I was there on the first day when Dr. Gourie-Devi entered and took a clinical case when she came for an interview. She came for an interview for the associate professor or additional professor level. At that time, she took a clinics and I attended the clinic as a non-course resident. And from that time, we are associated with each other for many, many projects. And she was the director and the vice chancellor from 1997 to 2002, heading this institute. Professor Gourie-Devi is a distinguished legend neurologist endowed with the qualities of astute clinician, enjoys a reputation as an inspiring teacher and a sought-after mentor to a large number of the students. As I mentioned, 5th generation of the students now, and has a passion for research even now. And she is doing actively involved even in number of research projects. She has made a notable contributions, and of which one of the most important is motor neuron disease. And she is credited with a description of monomelic AMI trophy, which she has coined the word. And in addition to ALS, her interest was varied, and she worked on Japanese encephalitis, clinical neurophysiology, peripheral neuropathy, leprous neuropathy, hyaluronidase in the management of spinal arthritis, neurotuberculosis, and neuroepidemiology. I was involved in the last 3 of her research projects. And this was the first lab of EMG lab, which we had in 1977 when she came here. The first time she started the DISA machine, EMG laboratory in 1979, and it was in a cage. You can see that cage even now. And this was one of the visitors from Nepal, the king, who was the visitor at that time. And this was Professor Baldev Singh, who was visited in our institute in 1986. Professor Baldev Singh was Dr. Gourie-Devi's teacher, and he was a professor of neurophysiology, very well known, and he was known as a father neuron in this country, and he was called as father neuron. And he was the teacher of Dr. Gourie-Devi, and that was the new laboratory. And this was a very important milestone in the history of NIMHANS that was conducted. This is the first international symposium of motor neuron disease, which was held in 1984. And this was organized by Dr. Gourie-Devi. I think all of us were there on the back. I don't think we can recognize even now. So half of us are not recognizable. And there are many, many dignitaries were there. The most important was that Professor Gajdusek, who was a Nobel laureate, was here, and Garoto was there. And then Professor Kurland was there. Hirano, you know about Hirano bodies. He was here at that time. Bradley was there. You must have read the Bradley's book also. He was here there. You can see that on the fourth. I don't think you can see here. This is Professor Bradley, and this is Professor Gajdusek. This is Professor Karlan and Dr. Nauzir Wadia, and then this is Hirano. And then we have Yase, and so many people were there, and we were all there on the back. Professor Chopra was sitting in the back, actually. So this was a great moment in the history of motor neuron disease, where we have all over the world, the people had come and seen here what was happening in 1984. And that started the momentum for the ALS work in this institute. And then her interest was varied, as I've said, neuroepidemiology. And here is Professor Dallas Anderson from NIH came here because I went to neuroepidemiology training way back. And at that time, and we were interested in neuroepidemiology and started the epidemiology here in this country. At that time, the Dallas Anderson was from NIH. I was in NIH for about 6 months. Then he came here and we started the neuroepidemiology survey in this country. And here is Aldous Huxley. I'm sure you all heard about Huxley. Aldous Huxley, eminent neurophysiologist, and Professor Desi Raju was a very well-known professor of neurophysiology in this country. I put it only because of the young people here who might not have heard about Professor Desi Raju. He was one of the great neurophysiologists of this country, and he was with us in this institute, Gourie-Devi and Desi Raju, interacting with Aldous Huxley, and he came here. And then the next one is Professor Walton was here. Sir Walton, you all know about Walton textbook, and Sir Walton was here, and we were all with him, and then he participated even in our clinical rounds. Next slide. And this is one of the important events of a brainstorming session. This was the beginning in 1984, one after another. We had a lot of events, and this was a brainstorming event which was held by Department of Science and Technology to take the neurosciences above the level. And that was the beginning of brainstorming session. And here is Professor, actually one slide behind. So the Professor P.N. Tandon, another teacher of Dr. Gourie-Devi's photo is there. Professor Ramurthy is there. And they were all great stalwarts of neurosciences and founders of the neurosciences in this country. Next slide, please. So this is one of the important events what Professor Gourie-Devi started was a neuromuscular clinic, which I'm sure many of our young people are working even today in the OPD and on the 4th or the 5th Saturday of every month. And this was a neuromuscular clinic initiated by Professor M. Gourie-Devi because of her passion and research in neuromuscular field. And this was set up way back in 1991. That means we are 35 years old, and this is still continuing every month, month after month, every last last Saturday of the month, which is continuing, and now it has evolved and with to genetics and various other things in a big way. And Dr. Nalini is leading that. Next slide, please. So this is one team, sir. This is known as ALS team. We had an ALS team here, and that team was led by Professor Gourie-Devi, and that is Professor Raju. I'm sure he must be on the online now. And Raju and Gourie-Devi, Nalini and Satyaprabha, they were meeting almost every week, at least once in a week, to discuss about ALS and how to take it forward. And the birth of that has led to the registry of MND in this country. And that is the one with Nalini has done that. Next slide, please. And Professor Gourie-Devi received number of awards, and I just want to put two of them. One is, this is Prime Minister of our country, Atal Bihari Vajpayee, giving an award called Om Prakash Basin Award in 1998. Next one. This is President Abdul Kalam was giving an award to Dr. Gourie-Devi, B.C. Roy Award. In recognition of her contribution, International Federation of Clinical Neurophysiology had conferred the prestigious Kugelberg Award lecture for an outstanding contributions to clinical neurophysiology in 2018. And Dr. Gourie-Devi has been very active in developing the field of clinical neurophysiology in the country from late 1980s. And it is gratifying that in 1990, in our IAN, there is a subsection of this. And there is a pioneering work she has done in neuroepidemiology. She was an expert member in many of the organizations, to name few: International Organization, World Health Organization, Fogarty International Brain Disorder Review Committee, World Health Organization Advisory Group on Revision of the ICD-10 Diseases of the Nervous System, and World Federation of Neurology. And national organization, I don't think there is any organization where she was not there. And ICMR, DST, DBT, CSIR, everywhere she is there. Even today, she attends to the ICMR as an advisor in many of the meetings, and I meet her very often. This is an important photograph. She became an administrator and heading the institute, and these are 5 sequential directors of NIMHANS, which are there at 150th year of our institute, which was held during the time of Dr. Gourie-Devi's period as the director. Next slide, please. And this was a farewell to Professor Gourie-Devi when she left this institute in 2002 after completing the term of the director of NIMHANS. And then this was whole of our neurologists from different parts. I think you can see many of us who are there. I think Tali is here and they're all there. And Dr. Nagaraj has not come, I think. So many of us are here in this group to witnessing the farewell of Dr. Gourie-Devi. Then this is, she came back and delivered another lecture on the overlap syndrome of motor neuron disease in the Department of Neurology. Next slide. So this meeting all the friends and family of NIMHANS in that way, and she had a lot of interaction with the people. Next slide. Yes, and this was instituted, Gourie-Devi's oration was instituted to carry on the legacy of Professor Gourie-Devi and her contribution to the neuromuscular diseases in basically. And this was the first Dr. M. Gourie-Devi oration. This was delivered by Professor Samir K. Brahmachari, who was the founder director of CSIR, IGIB, and former director general of CSIR. And that was a very interesting title. That evening at DMD clinic, this is nothing but neuromuscular clinic, what he was mentioning. He entered the neuromuscular clinic one day, and that has begun the research collaboration between Samir Brahmachari, that is from Indian Institute of Science, and NIMHANS. And that has taken in a big way. And that was the title he gave on the first oration in 2019. Next slide, please. Second, the oration was by Pamela Shah, Professor of Neurology, Sheffield Institute of Translational Neurosciences, harnessing the advances in neuroscience to translate into improved outcomes for patients with motor neuron disease. So in the same legacy, we are now moving on. Next slide. So third oration, and you are all here to witness this oration, and that is given by Professor Amar Al-Chalabi. And this title of the talk is very interesting. I'm very curious to listen to this talk, The Mystery of ALS. I was talking to him and say that we are waiting for a day when there is a treatment for motor neuron disease. Thank you very much.
Thank you, sir, for the excellent introduction. It was quite inspiring to listen to sir about Gourie-Devi ma'am and her contributions. He gave us a glimpse of the beginnings, the journey, and her immense contributions in establishing state-of-the-art facilities in the Department of Neurology in NIMHANS, as well as her contributions globally in the field of neurology and neuromuscular disorders, especially motor neuron diseases. I'm sure all of us sitting here are deeply inspired by her journey and her active contributions to this field now, even at this age, both clinically and for research. Thank you, ma'am. So now let me invite Professor Nalini, Madam, Senior Professor of Neurology, Dean Neurosciences and Lead Neuromuscular Specialist, to introduce our orator and to deliver the welcome address. Ma'am has carried forward the research work on ALS in India, which was started by Gourie-Devi ma'am, and she has contributed immensely to this field, and she has also received numerous awards, including Clinician of the Year Award in the last PACT ALS 2025. So let me invite ma'am to please deliver this welcome address. Thank you.
I don't need this. Thank you, Sina, and thank you, sir, for such a nice introduction on our beloved teacher. Ma'am, we can see you. You look the same as I saw you 25 years ago, and welcome. The same enthusiasm. I think ma'am has many more brain connections than any one of us. Thank you. So now, again, let me say a pleasant good afternoon to all our esteemed audience here, our esteemed orator, Professor Amar Al-Chalabi. It's a big name, so I'll just continue to tell us just Dr. Chalabi. Distinguished guests here, former directors, registrar, deans, medical superintendents, administrators, teachers, colleagues, residents, students, and friends. Yeah. Of course, it's again an emotional moment and lovely experience to organize this third Gourie-Devi Oration, where ma'am spent her life rendering selfless service for so many decades. Her passion for advancing neurology and improving patient outcomes, I would say, has made her an immortal, inspiring voice for the entire neurology community for generations to come. She set such high standards for patient care, training, and research, the legacy of which we are all really striving hard, but I'm sure that we can't match her legacy. Now it gives me immense pleasure, honor, and pride to introduce our most distinguished orator, Professor Dr. Amar Al-Chalabi. Professor Chalabi, my English may not be up to your British standards, but I beg your pardon in advance. So I truly don't know how to start and where to end an overwhelming CV that he shared, and justice cannot be done in a few minutes, but at least give me 5 minutes to do a little justice to what he's achieved. So he's currently the professor of neurology and complex disease genetics at the prestigious Morris Hall Clinical Neuroscience Institute, King's College London. And he's the head of the Department of Basic and Clinical Neuroscience and director of the King's Motor Neuron Disease Research Center. He's the co-director, UK M&D Research Institute, team leader for M&D NIHR Maudsley Biomedical Research Center. He's a senior investigator at NHR and deputy editor Brain. Please keep that in mind. He's the deputy editor of Brain, and he's promised that he'll help us with our submissions. Please do remember, Professor, all the promises that you made to us in the last 2 days. And King's Health Partners, and of course, consultant neurologist, King's College Hospital. So he's very well acclaimed, world-renowned and profoundly admired M&D specialist and complex genetic disorders expert, as mentioned. He's the chairperson of the Lancet Commission for Genetic Task Force, and he leads everybody by example and deep involvement. So he has held several important positions in the past, and just to list a few out of them: head of the Department of Basic and Clinical Neuroscience, and the program lead. He's also been the program chair for MSc in Clinical Neuroscience. That's very interesting. He set up the course, achieved university accreditation, and developed the full program between 2003 and 16. Congratulations, Dr. Amar. Instructor of Genetics of Complex Diseases, Cold Spring Harbor Laboratory, New York, conducted biannual 2-week residential course, visiting scientist at the Massachusetts General Hospital. We can only talk about these names, and I do not know how we even think that we could have reached these heights in one's lifetime. So he's also been an MRC clinician scientist at the Institute of Psychiatry. So his qualifications are incredible. So he's been the fellow of the American Academy of Medical Sciences. He's a diploma in statistics awarded with distinction, FRCP of the in molecular neuroscience. Is also a member of the Royal College of Physicians and completed his medical degree with distinction and 3 prizes. Congratulations, Dr. Amar. So let's give now and then. Okay, thank you. So Dr. Al-Chalabi is a renowned researcher, clinician, educator who works on M&D, and this has impacted countless patients and professionals. His passion for advancing clinical and basic research in M&D is awe-inspiring. His expertise and dedication to advancing neurological care have inspired many professionals, and students alike. I happened to closely interact with him during the FIGHT M&D roundtable meeting at Melbourne last year. And I have heard his lectures earlier and had great admiration for his extensive knowledge, experience, unique pattern of teaching. We all heard him this morning and how much enriched our knowledge became. And he was a simple person, mingled amongst us so well despite his great stature. And there's so much to learn from him and his deliberation. In fact, I was scared to talk to him at that FIGHT M&D, but now I'm no more scared of you. I'm going to talk to you freely. And one can't imagine his dedication and passion to find a cause and a cure for M&D as I walk. So a long day yesterday with him, presented a bit of our clinical palliative and research work being done. And it was so pleasing to see him engaging with each one of us and admiring our small work, and got deeply involved in the discussions and provided valuable suggestions to take our research work forward. And we were actually spellbound by his interest in our work. Really speaks of your keen interest in the subject, Dr. Chalabi. He completed his medical graduation and distinction from Leicester University, and subsequently he moved to London to do his PhD under the mentorship of a giant and veteran in the field of neurology, Professor Nigel Lee. Long back, I had dreamed that I should do a fellowship with him, but it did not work out. A leading specialist in M&D, and here Professor Amar's stupendous journey started with Professor Nigel Lee. And just after his PhD, he was awarded the prestigious Charcot Young Investigator Award, a rare feat, and this cemented his future in neurology, specifically genetics and M&D research. So he has a lot more to tell. He became the consultant at King's College in 2000, a research exchange scholar at Massachusetts General Hospital. I like telling that word, Massachusetts General Hospital, and Harvard University at Boston in 2001. He's very young, he's younger than me. And then a visiting scientist in neurology at MGH and the topmost biomedical universities in the world. This speaks volumes of his fierce passion about research interest. And as the genetic cause of ALS is still largely unknown and the prevalence is low, Professor Chalabi stresses the importance of international collaboration. And one such consortium is the Project MIND, for which he co-leads on a whole genome sequencing project aiming for 15,000 participants of ALS. We soon want to be part of this from NIMHANS also, along with Dr. Pamela Shaw, Dame, and Chris Shaw. And he also leads the European joint program for neurodegenerative disease research. I can go on, which looks at the development of personalized treatment by interaction analysis of various risk factors. And of course, with regard to his scientific research, he has a whopping 496 publications as on PubMed, which I searched, and the majority are in M&D dealing with clinical aspects, advanced research, genetics, and care. So let's give him a big hand for that. And just to name some of the top scientific journals that all of us have heard about: NEJM, Lancet Neurology, Nature Genetics, JAMA, Brain, etc., etc., etc. I can go on, but time is the limit. His collaborative research with another super giant in M&D, Professor Bob Brown, as we call her, Robert Brown at Harvard. So this is very important, which resulted in the discovery of the chromosome 9p linkage in ALS and frontotemporal dementia. So that's real achievement, Professor Chalabi, along with Professor Bob Brown. And then he performed the genome-wide association study of ALS with microsatellite markers. And it was an arduous journey, a convoluted method before single nucleotide polymorphism chips were commonly used for genotyping samples. But it allowed them to find a signal, and that was a breakthrough, an accomplishment he recalls with great pride. This speaks volumes with regard to his persistence in research. We congratulate you on the discovery, which has made immense change in the genetics of ALS and FTD. And of course, this is followed by his interest in complex genetic disorders. Another outstanding research, his team with Professor Christopher Shaw established that C9orf72 mutation that we all speak of it every day was a common genetic cause for ALS. And this was identified, and yet another landmark achievement by him. And then following this, he, along with his colleagues, identified new risk variants and 4 new ALS genes. And many congratulations to you once again. So he has several prestigious awards bestowed upon him over the years. So gold medal re-awarded for 5 years for the National Clinical Excellence Award. Forbes Norris Award for Compassion by the International Alliance. Healy International Prize for Innovation, Teaching Excellence Award, highly prestigious Sheila Essay Award for Research from the American Academy of Neurology. There are many more. I just thought I will just list out a few. And in 2021, he was appointed senior investigator at the National Institute for Health Research, a 1.2 billion British pound research budget, of which I think he's got about 50 million on paper and maybe around 21 million for research. And what he described to us was very touching. He had to take this campaign through 5 successive prime ministers of UK to get this approved. I think that was real persistence of yours. And I mean, I would say that you really literally stuck to it and got it done. Congratulations. And it also has a patent under review for low-dose human interleukin-2 for the treatment of amyotrophic lateral sclerosis in a subgroup of patients. Professor Chalabi, truly unbelievable and uninspiring, let alone daydreaming, maybe we can all dream of such achievements, maybe during our REM sleep only and not otherwise. So, and then morning we know that nothing is going to feel this thing for us. Early life, Professor Chalabi was born in Iraq. And when he was 2 years, he moved on to England. His father was a executive in the British Petroleum Company. And important for all of us to note that he moved several countries almost 5 times, relocating with his father's job, attending 7 different schools, and from which he laughs off, saying that it helped him to learn and get along with the diverse people and diverse cultures. So it's easy to talk to him. It's not, the typical I wouldn't say British, okay. Okay. Also interesting to note that he came for his electives to India in 1988 for 2 weeks, but spent 3 months. He's seen many more places than most of us sitting over here. And I felt a bit comfortable when he, coming from England, knew some things about India, its landscape, its people's behavior, and particularly the lack of civic sense. So he's got used to the lack of civic sense in India. Thank you for that. Okay, last but not the least, many instances when doctors become leaders in their field and win prestigious awards by sheer persistence, and he's one of them. From a very young age, Al-Chalabi was intent on practicing medicine, aiming for heights of the Australian Flying Doctor Service, and his resolve to get into medical school after several attempts, and he was absolutely successful. Many lessons to learn and carry from you, Professor Chalabi. Now I would say you, Sir Chalabi. It just means that we need to be at the right place and among the right persons and with the right motivation that we could reach great heights like what he has achieved. And intriguing, last one, there was a nice document written in Lancet Neurology by Jules Morgan. And he says that Chalabi, if he's not found in the lab or in the clinic, he will be at his home with his wife and 2 sons. And if he's not there either, he's jigging. And for the past several years, he has been drumming in a band. I wanted him actually to do the drumming today, but we didn't get a proper drum for him. Acoustic rock, Eagles crossed with Rolling Stones is how I hear it, although the guitarist would probably disagree. So he's a rock star actually in many guises. And today we have the rock star who's going to rock in motor neuron disease with us. And he always says that patients motivate him. And he says without any hesitation, I want a cure for everyone, a treatment that stops the disease or reverses it, and hope that day will dawn soon and all MND patients will see the light of the day. So, Professor Al-Chalabi, we are all delighted to welcome you today to deliver this prestigious M. Gourie-Devi Oration, our beloved teacher. And now the podium is yours. Sina, would you like to? And the podium is yours. Let's start.
Thank you, ma'am. So we were wonderstruck by his achievements, and as ma'am mentioned, his hard work, perseverance, and persistence has led to immense contribution to his research and care of patients with motor neuron disease. And I know that we are all eagerly waiting to listen to him. So without any further delay, let me invite our distinguished orator, Professor Dr. Amar Al-Chalabi, to please come to the podium and to deliver the oration. We welcome you, sir.
Thank you very much, Sina. And thank you, Professor Nalini and Professor Sathachandran. And I'm very honored to be able to speak to you about the mystery of ALS. Will it ever be solved? For the 3rd Dr. M. Gourie-Devi Oration. Thank you for inviting me. So, I know that not all of you will be clinicians, so I thought I'd start by just explaining what ALS is from a clinical point of view. So here you can see a cross-section of my brain. This is my MRI scan. And you can see on it that there is this red neuron. That goes from the cortex down to the bulbar region, which is the medulla, or it can go all the way down into the spinal cord. And there, that neuron talks to another neuron, which you can see in yellow, which goes out to the periphery and engages with the muscle. And in human beings, we are almost unique in the animal kingdom in having such a simple motor circuit. Most other animals have one neuron talking to another neuron, talking to another in a big chain. And at each point, they will diverge and speak to many neurons. So for example, if you're a mouse or a rat, you can't do a simple pincer movement because you don't have the one-to-one mapping between the motor neurons in the cortex and the muscles in the hand. You can only make a very large grasping motion because of that diffusion. So this means we have the most advanced motor system in the animal kingdom. We have this ability to speak, to move in a very, very coordinated way. We can have people on motorbikes in a pyramid and the person on the top is juggling flaming clubs. No other animal could do such a thing and do it so diversely. But that advancement, that, if you like, it's like a Ferrari motor system, it has its downside. Like a Ferrari, which is a very powerful car, it's also very difficult to maintain. And if it goes wrong, then it's very difficult to repair it. And our motor system is similar. It's a very advanced motor system, but if it goes wrong, which it's more prone to, then it's more difficult to repair. And what happens is quite characteristic. So if the upper motor neuron is damaged, the person will develop weakness because they can't get the signal from the cortex to the muscle. But that weakness is a very stiff type of weakness because one of the roles of the upper motor neuron is to relax the muscles. And when we test the reflexes by tapping the tendons, they jump a lot because it's part of the same circuit that's relaxing the muscles. So if that's not working, the muscles will jerk. If you damage the lower motor neuron, then again, there's a break in the circuit from the brain to the muscle, and so there'll be weakness. But that neuron releases trophic factors to keep the muscles large. So if those are gone, then the muscle will waste. And you may see muscle twitches as the body tries to repair itself, called fasciculations. And there'll be a loss of reflexes because the lower motor neuron is in charge of the reflex loop. So when we see that pattern of stiffness and muscle wasting, and we know both the upper and the lower motor neuron are affected, there's not really many other conditions that can do that. And especially if we see that in someone who's 50 or 60, it's very unlikely the diagnosis will be anything other than motor neuron disease. It's nearly always motor neuron disease in that situation. And this is what it can look like. So this was one of the first people I saw actually back in 1994 at King's. And she has, as you can see, her tongue it's frozen by the flash. It's lumpy, and that's because it's twitching. And it's more difficult to see on the camera, but it's smaller than it should be and thinner. And it's also coated in white. And that white is thrush, a fungal infection, because she can't move the tongue around the mouth and therefore can't prevent those kinds of infections. And you can see there's saliva around the tongue, and her mouth is more moist than it should be. Again, it's more difficult to swallow if your tongue doesn't move properly. And so that's another problem our patients have. Here you can see what happens to somebody's arms. This man has very, very thin waisted shoulders. He looks like, it's called a man in a barrel. It looks like somebody stuck in a barrel. And here you can see the woman's hands, the same woman with her mouth open. And you can see, if I just point to, can I go back? Sorry, thank you. If I just point to this part of the hand, you can see at the base of the thumb, there's a lot of muscle loss. And also between the thumb and the index finger, there's a lot of muscle loss. There's muscle loss in the other places as well, but those two are very characteristic of motor neuron disease. That's called the split hand. For reasons we don't fully understand, but are probably to do with the cortical wiring, the side of the hand closest to the thumb is more likely to show weakness before the other parts. You see similar patterns, for example, the biceps tends to be weaker than the triceps for similar types of reasons to do with the wiring. Now, there are two obvious causes of ALS. So my question at the beginning, what's the mystery of ALS? Will it be solved? Well, we know there are at least two causes. So my slides are projecting a little bit strangely, but hopefully it'll work still. So the first is that family history is present in 5 to 10% of people. And so that tells us that genetics must be important in ALS. So that must be one cause at least. And we know that the risk increases with age. The older you are, the more likely you are to develop ALS. It's rare, apart from Madras MND, it's rare for somebody to be less than 18. In fact, very rare, even under the age of 30 or 40. But when you reach your 50s, 60s, 70s, it becomes more frequent. So that tells us that whatever is causing ALS, the mechanism must involve something to do with aging or being older. So that's a mystery that I wanted to just store in your head. So if we look at the family history part of it, we know ALS is genetically complex. What you can see here is a pedigree from a family with a single gene cause. So if you're not familiar with pedigrees, they're not usually in a circle like this, but you can imagine this if you stretched it out horizontally. So the middle line, the centermost line, there are two people just here. These are the initial founder parents. So every time you go down a generation, you get another ring, and anybody who's affected is colored in black. And you can see there are various black dots scattered around this. Roughly 1 in 10 people in this pedigree is affected. And you can see the part that I've ringed looks like familial ALS. So there's a grandparent, then there's a father and son affected. So if you saw that person in clinic, if you saw the youngest person there, it would look like they have a family history of ALS. But if you were to look at some of the other people, for example, down here, these are fifth cousins. And most people don't know their fifth cousin. So if one of those people came to your clinic, you would think they have sporadic ALS. We actually know in this pedigree, they have a variation in a gene called C9ORF72, which in many parts of the world is the most common genetic cause of ALS. But carrying the gene isn't enough to develop ALS. We don't know why. About 45% of people who carry this gene variant will develop ALS by the time they reach their 70s or 80s. But not everybody does. Some people develop frontotemporal dementia. Again, we don't know why they develop that instead of ALS. Some people develop both. Some people have nothing. Some people develop schizophrenia or Parkinson's. So what's determining those things from that gene? Why do some people develop ALS and some people develop something else? So that's another mystery I'd just like you to hold in your head. So what about genetic testing? So we know, we did this study in 6,000 people with ALS. If you screen for 24 genes known to be important for people with ALS, then the rate of finding a positive genetic result that you would act on clinically is about 20%. And it doesn't matter how old you are when you're tested, everybody has a risk of about 20%. In fact, our more recent study shows it may be 25 or even 30%. So this shows that gene effects, these are people with no family history, that genetic risk factors are important even in people with no family history. If you were at the talk this morning, you'll know why that might be. And if you're more than 70 years old, then the risk drops a little, but it's still significant, 15%. So that means we should offer genetic testing to everybody, regardless of age, regardless of family history, because a hit rate of 20 to 30% is very important. It has implications for other family members and potentially for treatment, because there are some genetic therapies now. Now, one thing that we often see is that people who come from a family with ALS tend to develop ALS 5 to 10 years younger than people who don't have a family history. And the question has always been, is this because they have ascertainment bias? If you're aware of the symptoms of ALS because other people in your family have been affected, maybe that's why you'll detect the symptoms earlier and you'll present earlier. And maybe that's why they seem younger when they come to the clinic. Or is it some other thing? Is it having a genetic factor that lowers your age of onset? So we thought we would try and investigate that. This was one of my students, Pooja Mehta, who did this. So we looked at 100 people with familial ALS and 840 people roughly with apparently sporadic ALS, so no family history. And we found indeed the people with the familial ALS were 5 years younger at onset, and that was statistically significant. But if you remember, I told you that you can carry a gene for ALS even without a family history. So we could split our people with sporadic ALS into two groups, the ones who have a known genetic factor and the ones who don't. And now we can answer the question about ascertainment bias. Because if the people with a genetic risk factor have no difference in their age of onset, but they do have an onset younger than the people without a genetic risk factor, that tells you it's genetics. Whereas if they are still younger, if the green and the blue match on the right, that tells you that it's ascertainment because having the family history is what matters. And actually, when we do this, we see that there's no difference. Unfortunately, the way the slides have, I think in transferring it to the Mac, it's adjusted all my fonts and everything, so I'm sorry about that. But there's no difference. Actually, the p-value is, I think, 0.2 or something like that. There's no difference in age of onset. But if we look at the genetic versus non-genetic, then they are younger, and that's statistically significant. They're younger by about 3 or 4 years. So that tells us that actually carrying an ALS gene in itself lowers your age of onset. We don't know why that might happen, but carrying the gene itself is enough to give you a younger age of onset. But if you remember my slide where I showed you it doesn't matter what age you are when you're tested, it still doesn't matter from that point of view. You should still test everybody. Now, I've talked a lot about risk of ALS, but if we're trying to develop a treatment, we don't really care about risk, except that it might tell us something about the mechanism. Because when we're trying to treat somebody, they've already got the disease. We already know they've had the risk. They've got it. What we care about is the progression. Can we slow the disease or stop it? So it's important to understand what affects how fast your ALS progresses. Some people will die within 6 months of their first symptoms. Some people, like Professor Stephen Hawking, lived 50 years with ALS. Most people are dead within about 2 years, but about 20% of people will live 5 years, and about 10% will live 10 years. So there is quite a distribution of survival. Why are some people protected and have a slower disease than others? It's a very important question. And it also matters if we can find the variants that affect, presuming it's genetic, how fast your disease is progressing, you might be able to make a drug that targets that in everybody and therefore slow the disease down. So we looked at one gene where we have a lot of information, and that is the SOD1 gene. And here you can see on the x-axis many different variants of SOD1 that are plotted. And on the y-axis is the log of the disease duration in months. And I've put 2 thin blue lines. I don't know if you can see them. You should be able to. Unfortunately, as I said, the font is adjusted in the screen width and things. But sorry, could you go back before that? Thank you. But the bottom line is a 2.5-year survival, and the top line is a 5-year survival. And then if you have a look at the A5V variant, which is the one that's leftmost, where it says very aggressive, this one, you can see that most people carrying that variant have a survival of less than 5 years. And if you look at the less aggressive one here, D91A, most people there have survival more than 5 years. So there's a relationship in the same gene between different variants that affect your survival. But then if you look at this other one, I114T, you can see that there's a very wide distribution of survival, and it doesn't seem to matter having that variant. Some people live a short time, some people live a long time, and many people live in between. So even within the same gene, different variants are associated with different survivals. So it may be something to do with the mechanism of how those variants work. But we must assume, since they're in the same gene, that the mechanism is similar for each. So it's still not clear why this happens. Now, we know that large effect Mendelian disease genes can cause ALS. I showed this slide this morning. This shows you that on the right, I've got all of the different genes discovered for ALS up to 2017. Because there are so many, I didn't fill the whole curve. If we carried on till now, it would be huge. But roughly, you can see the number of genes discovered is doubling every 4 years. I've also colored the spheres by the number of publications for that gene. So the large spheres have many publications. They must be important for the field. And they're colored red or blue. Red is where they're found in people with sporadic ALS, no family history. And the blue is where they're found in people with a family history of ALS. So again, you can see having a family history or not doesn't really affect whether it's a genetic cause. And you can look at the pathways these genes are involved in, and you can see there are at least 3 pathways. The green are involved in protein homeostasis. The bluish purple are involved in altered RNA binding proteins and RNA processing. And the orange-yellow ones are involved in cytoskeleton proteins. So those 3 mechanisms, those 3 things must be important for ALS. And these different variants are cataloged in an online database called the ALS Online Genetics Database. But also, many people publish saying this is an ALS gene. So we have to know, is it really an ALS gene or not? And the way we do that is we have an adjudication committee that looks at the evidence, and we decide for each one, what's the evidence systematically? And then we rate them as definite, limited evidence, strong evidence, whatever it may be. And then within each one, we ask, is that variant a variant of uncertain significance? Is it a pathogenic variant, or is it a benign variant? So we have mutation adjudication as well. And this information is all available publicly online. So is there a common mechanism for ALS? So everybody with ALS, except for the 2% of people who have SOD1 mutations and the 1% of people who have FUS mutations, everybody else, doesn't matter if it's genetic or not genetic, has an accumulation of this protein TDP43 in the cytoplasm. And that protein should normally be in the nucleus. TDP43 was initially discovered during HIV, when the HIV pandemic was upon us. It inhibits retroviruses, but it sits on things called cryptic exons. So in your exons, when you're transcribing them into protein, some of the introns contain an exon as well. But that exon is switched off by TDP43, so it doesn't get turned into protein. But if TDP43 is in the wrong place, if it's in the cytoplasm instead of the nucleus, and it's lost its function, then those cryptic exons are unmasked, and the proteins that are made are either truncated or may have abnormal sections in them. And so you can imagine that that's also making an antigenic profile. It means you're exposing different antigens to your immune system that you wouldn't have normally seen. It also disrupts the transcriptome in the proteome. So it really has moved all my slides and pictures around. So the heritability of ALS, you can ask, how much, I've shown you the Mendelian risk, but what about the small risk factors? What's the heritability of ALS? What's the chance of small genetic risk factors nudging you towards getting ALS? So the way you can answer that is you look at twin studies. Identical twins, because they share all their genes and all their environment, they should, in a genetic condition, match 100% or close to 100%. And in a non-genetic condition, then they would match more like non-identical twins who are just siblings. So in ALS, we looked at 49 identical twins and 122 non-identical twin pairs. And we saw that for the non-identical twins, in none of the cases did both twins have ALS. But in the identical twins, out of 49 people, 5 of them, both twins were affected. And if you crunch the numbers and do the maths, that tells you that the narrow sense heritability, that's the amount of genetic variation contributing to the phenotypic variation, the risk of ALS, is about 60%. Now, we haven't been able to explain all of that 60% yet, even with all of our studies, which I'll show you in a moment. But it does tell you that's the target we need to look for. So one of the first studies we did was a genome-wide association. We looked at 27,000 people with ALS and 111,000 nearly controls. And we tested them for about 10.5 million genetic variants. And then from there, we imputed more, up to about 100 million. And what you can see on the graph is on the x-axis, every chromosome, and on the y-axis, the minus log 10 of the p-value. So the higher the dot, the more significant is the association at that point. And every single association, every one of those 10.5 million variants is plotted as the result of its chi-squared test. Is it more frequent? Is that variant more frequent in cases than controls? And the p-value is shown there. And you can see immediately the very large landscape, the very large skyscraper, sorry, in the middle, C9orf72. As I told you, that's a very common cause of ALS internationally. And you can see these other variants also contribute. And if you look at the pathways these are in, they implicate cholesterol and the immune system. So that may be a bit surprising. Why should cholesterol matter in ALS? And why should the immune system matter? Because they're motor neurons, they're in the brain. Why would they be affected by the immune system? We've also done an exome-wide association in ALS. If you just look at all genes in general, then again, we've done the same kind of thing here. We've just looked at all the rare variants in the exome. The orange ones here are genes that have not been previously identified as ALS risk genes. So this came out this year. But again, it just shows you we've then got 1, 2, 3, 4, 5 new genes just like that. We're discovering many genes associated with ALS all the time. And then if you look just at the ALS genes, so if you remember, I told you about that gene curation expert panel at NIH, the ClinGen GSEP. So in that panel where we rate the different genes as definitive evidence, strong evidence, etc. So these orange ones here are genes that were previously regarded as having limited evidence as being ALS genes. But now we have very strong evidence. So when we reevaluate them, we'll move them up to more important genes. Now, one of the things we know is that if you have a new mutation, if it's benign, then there's nothing to stop it being selected against in the population. It could drift to high frequencies. But if it's a deleterious mutation, then in general, it will be selected against because it's deleterious. You'll have fewer offspring or fewer offspring that reach reproductive age. And so if you find a variant that is rare, it's more likely to have a big effect on your disease process. And if you find a variant that's common, it's more likely to have a small effect. It's more likely to be towards the benign end. And these are all the variants we've seen in ALS. You can see, in general, the slope follows that rule. So the big effect size genes at the top compared to the small effect size genes at the bottom, the small effect ones are commoner. It's really lost all of my graph pictures. It did seem to project before when we were practicing, didn't it? I'm sorry about that. It's Windows, yes. Yeah. Yes. It's changed. I mean, it's changed I usually use Arial font. It's changed it to a kind of courier font. So I don't know why. Anyway, it doesn't matter. I'll just carry on. I can talk through the problem.
Can you hit the slide change it to say next instead of the
It doesn't do it. Okay, thank you. Is it working now? Yep. Nope, it doesn't matter. If you could go back one slide. Thank you. Okay. If you could go back one slide, please. So if we go back to our two obvious causes of ALS, we've covered the genetic family history side, but the other one is age, which tells us the mechanism must be something to do with age. And if you look at the lifetime risk of ALS, you might think of it as a rare condition. In Europe, at least, the incidence, and this seems to be true worldwide, is 1 to 2 per 100,000 person years. So that means if you observed a population of 100,000 people for a year, you'll see 1 or 2 cases. So it seems rare. But actually, the lifetime risk is 1 in 300. So if you're on an airplane with 300 people, one of them on average will develop ALS. At your school, if there were, I don't know how many people are in a typical school in India, for us, it's maybe 800 people. So 2 or 3 of those people will develop ALS in their lifetime. So it's much more frequent than you might imagine. And what you can see on this graph, so the x-axis should just be every notch is a year up to 90. So it's kind of stretched them out for no reason. So the female line is the black line. You can see women at every age have a lower risk of ALS than men. The ratio is about 3 to 2 male to female. And you can also see that the older you get, the risk just keeps increasing. It tails off as people die as they get to the 90s, but the risk just keeps increasing. So maybe if we all live to 200 or 300 years, maybe the risk would be close to 100%. Maybe we all get ALS if we live to 1,000. So this is an intriguing finding in itself, that the risk just keeps increasing. It's not that it peaks and then drops off. So we can model ALS. We can imagine that everyone is born with a genetic load, a genetic predisposition, and that takes you towards a threshold, which I've drawn with this red line here, onset. And they develop cell damage with time. And then at some point, they have environmental exposures that also may take them towards that red line. But if you don't reach the threshold, you won't develop ALS. But you can imagine, so sorry, this is GTE, gene time environment. You can imagine that in some people, they accumulate enough environmental or time or genetic damage that at some point they reach that threshold, and then they will develop an inevitable decline to death. Okay, so you can model ALS like this. So you can imagine there's a burden of disease-causing factors. We don't have to know what they are, but we just can see this. So I want you to hold that model in mind. Now, if we look at environmental risk, lots of people have looked for environmental risk. Now, the genome, 3 billion base pairs, and you're looking for 1 or 2 gene variants, it seems like an impossible task, but it's achievable if you get large enough numbers of people and you do enough technology and assessment, you can find the genes. Environment is worse. It's much more difficult because it could be anything. Maybe you ate an apple seed when you were 3 years old, or perhaps you lie with the pillow pressing on your carotid artery too much, or maybe you get too hot in the shower. It could be lots of things that we don't test or we can't test that would be causing ALS. So people look at the obvious causes, but we could still miss the cause. People have looked at military service, and there is some evidence that having been deployed in the military to a war zone and then come back might increase your risk. It does seem to be a replicable risk factor. It probably doubles your risk, something like that. Pesticides, some people have shown that pesticide exposure might increase your risk. Other people have shown no increase with pesticides. Heavy metals, the same kind of story. Some studies show an increased risk, others show none. Smoking, there have been something like 50 studies of smoking in ALS. Half of them roughly show that smoking increases your risk. The other half show either it has no effect or don't provide sufficient evidence. So again, inconclusive results. Exercise is a particularly challenging one. So in the UK, we have many rugby players and some football players affected with ALS. And so there's a lot of interest now in whether rugby or football increase your risk. There was a study from Italy that suggested professional football increases your risk 6 times. If you analyze the same data another way, it doesn't increase your risk. Rugby, when we raised our £50 million from the UK government, part of our campaign was 2 rugby players and a footballer who went on to the BBC Breakfast News every day, not every day, every 3 months, roughly, and showed themselves to the public deteriorating and saying, we need an effective treatment. Rugby is very strongly in the minds of people associated with ALS. But exercise, if you do the epidemiology, there's no strong link between exercise and ALS. And part of the problem is, I showed you that cholesterol is implicated in those pathways. And we know cholesterol causes heart disease, raised cholesterol. So if you imagine people have raised cholesterol, it increases your risk of heart disease and ALS. If you exercise, you're protecting yourself against the heart disease. And now the people with ALS are the ones who've exercised because they were protected against the heart disease. So it could be something like that. Or it could be, I showed you earlier that we have a very advanced motor system. People who are athletes, maybe they have an even better motor system. It's even more vulnerable than the people who are not so good at these sporting activities. And that's why they're predisposed. It doesn't matter if they exercise or not. It's having that background that makes them skilled sports people. So even if there was an association between exercise and ALS, the actual message we give people, we have to be very careful about, because exercise is so important and it reduces your risk of so many other conditions that are much more frequent than ALS. But the bottom line is that environmental risk factors, there's variable evidence and they're difficult to replicate. So the title of my talk is about ALS mysteries. I want to list the mysteries. I've given you some of them already to hold in your mind. So the first is that ALS genes also cause other diseases. So quite a few ALS genes increase your risk of frontotemporal dementia as well as ALS. The second is not all gene carriers develop ALS. Why is that? You carry a disease gene from birth, then you'll have nothing, and then when you're 50, you suddenly develop ALS. Why is that? Or you reach all the way to 90, you never developed it. What's going on there? I showed you that there's an increasing risk with age. Why is that? And I also, I haven't shown you this, but it's implied, there's age-dependent penetrance. So if you carry a disease gene, like C9orf72 or SOD1, you don't usually develop ALS when you're an infant. You develop it in adulthood. And the older you live, the more likely you are to develop it. I showed you that age curve. So that tells us that the probability of expressing that gene variant increases as you get older. So that's age-dependent penetrance. I've just shown you environmental risk factors have been very difficult to find. Why is that? There doesn't seem to be a single cause of them. So those are the ALS mysteries. So I'm going to propose to you at least one component of a solution, which some of you I know at least have seen before. So I should point out that's similar to cancer. Okay, all of these things are similar things in cancer, age dependent, that genes can cause other conditions, etc. So in cancer, in the 1930s, two scientists, statisticians actually, Armitage and Doll, developed a multi-hit model of cancer. And in that model, if you do the maths of it, which I'm happy to go through with other people outside this talk if they want, if you do the maths, you can show that for a multi-hit model, there should be a power relationship between the age of onset and the incidence. And the power, the actual exponent of that power relationship will be proportional to the number of steps. And what that means in practice is if you plot the log of age on the log of incidence for a particular condition, then if it's consistent with a multi-hit process, a multi-step process, it will be a straight line. And the gradient of that line gives you information about how many hits are involved. Okay. So for ALS, we did this. And we looked at the Southeast ALS register, which is the one I was running from King's at the time. And you can see on the y-axis is the log of the incidence, and on the x-axis is the log of age. And you can see it's consistent with a straight line. The R-squared is 0.95. So that shows that at least in our Southeast ALS register, southeast of England, that it was consistent with a multi-step process. It doesn't mean it is, but it means it's consistent with it. We then went to our colleagues in the Netherlands, who also had a population register, and they had a very nice straight line finding with an R-squared of 0.99. And then we went to colleagues in Italy, in the Piedmont register, also an R-squared of 0.95. In Ireland, R-squared of 0.99, a very nice straight line. And then if you look in Scotland, 0.97. So all of these different population registers are all saying ALS is consistent with a multi-step process. Now, if you combine all of them, it's 0.99, and that tells you they all have the same gradient. They're all saying the same message. Okay, and if you look at the gradient, then it's about 5. And since the gradient in the maths is 1 less than the number of steps, that tells you ALS is consistent with being a 6-step process. It doesn't tell you what the steps are, but it tells you that 6 molecular steps could be causing ALS. Okay, so that's one possible explanation for ALS. And it's quite a nice explanation because it resolves many of these ALS mysteries. So the fact that ALS genes also cause other diseases could be because if you have steps 1, 2, 3, 4, 5, 6, you develop ALS. If you have 1, 2, 3, 4A, 5, 6, you develop frontotemporal dementia. So you have a slightly different path. Not all gene carriers develop ALS because the genes won't account for all 6 steps. Maybe they'll account for 3 or 4 of them. I'll show you some evidence for that in a moment. And then if you don't have the other triggers for the other steps, you won't develop ALS. But having the genes will greatly increase your risk because it takes you a long way towards the 6 steps. And that's why genetics increases your risk, but it doesn't make it 100%. The increasing risk with age is because it takes time to accumulate these hits. You're not born with the 6 hits. You have to take time to get them. There's a small probability per year for each one. And the age-dependent gene penetrance for the same reason. It just takes time to accumulate the hits, even if you carry a disease gene. Environmental causes are difficult to find because these are molecular hits. And you might have multiple causes for the same molecular hit. In the same way as smoking is carcinogenic, but so is radiation and traveling in an airplane. If you have a relatively rare condition, it may be difficult to pull out the different environmental risk factors that are all causing the same molecular step. And there's no single cause because different factors could trigger the same molecular step, the same kind of reasoning. So having this multi-step model, I can't say it is a multi-step model, but it's consistent with a multi-step model. And it would resolve these mysteries quite nicely. And this finding has been replicated by many others. So in the Australian register, it's been replicated twice, in the Danish register, in a Japanese, and twice in an Italian register. So it does seem to be consistent. South Korea, when they replicated it, they showed it was a 5-step process, which suggests that there is some predisposing risk factor in South Korea that already accounts for a step. They have a greater risk. on a population level. But the key feature is that more steps is more protective. Okay, so if you see fewer steps, you've got greater risk. If you see more steps, you're protected. So one of the things we did was we looked at genes. So we were able to do the incidence and prevalence with our Italian colleagues because they have genetics on everybody. And we could see that if you carry the C9orf72 gene expansion, that accounts for 3 of the steps. So you've only got 3 left. If you carry the TDP43, the TDP43 gene is TARD-BP. If you carry that, it's a 4-step model. So it's slightly less risky because more steps is protective, but it's accounted for 2 of the steps already. It's nudged them a long way towards the risk of ALS. And if you have SOD1, which is generally a high penetrance gene variant when you have it, that accounts for 4 of the steps already, and then you only have 2 more to get the disease. So genes do account for some of those steps, but not all of them. And lifestyle can also account for some of those steps. So this was a study from Australia where they looked at ALS. They again showed 6 steps. But if they looked in people with ALS and heart disease, they showed 7 steps. So it looked protective having heart disease. But actually, when you take into account, I told you already about the competing causes of death. When you take that into account, actually, it becomes a more risky thing. It's 3 steps. So you do have to do the statistics carefully. But heart disease somehow is involved with ALS risk. Why is heart disease associated with ALS? Well, one of the things we did from our genome-wide association study is a study called Mendelian randomization. So to understand this, it's quite simple. Imagine you were completely unethical and you wanted to discover, does smoking cause cancer? What you could do is randomly assign people at birth, and half the population, you say, you must never have a cigarette ever in your life. You're forbidden. If you ever have a cigarette, you'll be severely punished. They're never allowed to smoke. And the other half, you say, you must smoke. You have to have at least 20 cigarettes a day for life from childhood. Okay? If you do that, you would very quickly discover, it's a randomized trial essentially, you'd very quickly discover that smoking is associated with cancer, and it's causal because you've done a randomization at the beginning. Okay? So it's not just an association, it's causally related. Now, we do have that process essentially in the form of Mendelian randomization. When you get your genes from your parents, they're randomly assigned to you from each parent. And so you can use the same kind of statistical logic. It's like a clinical trial. If there's a variant that increased your risk of smoking, and there are in the acetylcholine receptor, if you have variants that increase your risk of smoking compared to variants that don't, you could look to see, do those variants increase your risk of cancer, even though they're not directly causing cancer? And you can show that they do, and therefore there's a causal relationship between smoking and cancer. So we've done similar kinds of things in lots of diseases. This was a study in ALS. So we looked at the genes controlling your total cholesterol level, the gene variants. And if you do that and do the maths, you can show that total cholesterol is causally related to the risk of ALS. And it's not the reverse, because you can look for reverse causation with this method. Having ALS does not change your risk of what your cholesterol level is. Okay? So that immediately tells you cholesterol is somehow important. We don't know how yet, but it is important. You can also look with epigenome-wide association. So the epigenome that we looked at here was methylation status. So CPG islands in your DNA are methylated, and that can regulate genes. It can switch them on or off. And we looked in the same population that we did the genome-wide association study in, and we could see various pathways. So the blue represents the cholesterol pathway. It's implicated in ALS from the epigenetics as well. But also I want you to see inflammation is important. So inflammation, why is that important in ALS? We've done cluster analysis of the brain transcriptome. So we have roughly 250 brains available to us in the UK from people with ALS. We took from the motor cortex samples, and we did this as well. We replicated this study from multiple other data sets, and we also did it in blood. And if you simply just do cluster analysis, a statistical method, you can show that there are 3 ALS subtypes. There's one involved in synaptic and neuropeptide signaling, one involved in oxidative stress and apoptosis, and another in neuroinflammation. What's not clear is each person just in one of these clusters when they first develop ALS, or do people start in one? So maybe you start with a neuropeptide problem and then you migrate into the oxidative stress group and then you become neuroinflammation towards the end. Or maybe you're always in all three groups, but one is predominant compared to the other. We don't know yet, but these three clusters you can replicate, it's been replicated by others, and you can classify people in this way, not just from brain biopsy, but also from blood transcriptome. And again, neuroinflammation is one of the clusters. And that brings me to the work of Professor Raju, and which Professor Nalini was involved with, and Anu Mary Varghese, and which was presented to me yesterday by Vijay Lakshmi, which is on chitotriosidase, which is a very nice biomarker. If you inject it into into cell models, it will induce a motor neuron disease-like phenotype in those models. So it looks very powerful. And you can see here, it also looks as a biomarker. If you look at the bottom, for example, in panel E, so the spinal fluid of people with ALS is on the right, and the spinal fluid of healthy controls is on the left. You can see there's almost none in the healthy controls and a very big amount in the people with ALS. So it's very nice. There have been 46 papers subsequently looking at this from across the world. So inflammation in ALS, we know CHIT1 and other molecules will activate the macrophages and microglia in ALS, and they reduce the T regulatory lymphocytes, which are like the police of your immune system. Lower levels of T regulatory lymphocytes are seen in people with ALS, and they're also dysfunctional. And the lower the level, the faster the disease progression. So we just did a clinical trial of low-dose interleukin-2, which is a molecule that will increase the number of Tregs and increase their function. And we showed that it improved survival in people with ALS. It was a phase 2b study, but it was about 220 people in that trial. So now we're going to do a phase 3 study. So the multiple steps, what could they be? Could there be some kind of immune genotype, some inflammatory trigger, an inflammatory response? What could be going on? That should say retroviruses and ALS. So 59% of people with ALS have a retrovirus signature in their blood. They have reverse transcriptase in the blood, compared with about 5% of the general population. And relatives have the same frequency as patients, whereas spouses don't. So that tells you it's not a sexually transmitted retrovirus, it's an inherited retrovirus. Okay, and that may surprise you that retroviruses can be inherited, but actually 8% of your genome is inherited retroviruses. 1% codes for your protein, 8% is these retroviruses. And that's because if you think how a retrovirus works, when you're infected with a retrovirus, its life cycle involves being converted into DNA and going into your genome and then being transcribed back into a retrovirus. So it's designed to jump into your genome randomly. And that means if that happens in your germline, that's transmitted to the next generation. But it also means when that is in an adult body or in a developing person, once it's in a human, it can be expressed and then it can go back into the genome in another place. So you can end up with multiple copies. And through evolution, these become degenerate and mutate and disappear. So in humans, we don't have any functioning full-length copies of retrovirus, but we do have bits that are important. Now, these are all called HERV, human endogenous retrovirus, and then they've got a letter after them. So HERV-F, for example, is crucial for the formation of a placenta. All mammals have HERV-F. If you don't have it, you can't make a placenta. So they're not just bad, some of them are good. And HERV-K appears to be the source of this signal of retroviruses in humans. And HERV-K is the most recent retrovirus to become incorporated. It's something like 270 million years old. And there are sequences of HERV-K, although the whole thing isn't in one segment, all the different bits exist. So you could, in theory, assemble HERV-K. We don't, but we know that the envelope is toxic to motor neurons specifically. So it may be, remember TDP43 that's in the wrong place and is controlling retroviruses? Maybe one of the things it's doing is unmasking these retroviruses. So then they're expressed and HERV-K envelope protein is expressed, and then that's toxic to motor neurons. Maybe. So one of the things we did is the Lighthouse, sorry, could you go back? Thank you. Is the Lighthouse 2 phase 3 clinical trial, where we used an antiretroviral to see could you treat people with ALS and prevent it. Now, that trial took on 410 people. In the end, we stopped it for futility because we didn't show a difference between the groups. It doesn't mean the retroviruses aren't a good target. It may be the drug we chose wasn't the right drug, or that the mechanism we used to target it wasn't right. But we know that that particular approach didn't work. But I want you to also just look at this, which is a motor neuron disease-like disease in mice. So these are naturally occurring mice. They're not laboratory mice. And some mice are born with that. They have their own endogenous retroviruses called murine endogenous retroviruses, MERVs. Some mice have many copies and other mice have not many copies. And some mice have a gene variant that means they switch on their copies. Other mice don't have that. So if you happen to be a mouse with many copies of endogenous retrovirus and the gene variant that switches it on, and then you're infected with murine leukemia virus, you'll get a motor neuron disease. So as a model, it's quite interesting because it combines the genetic risk factor, which is the genetic background, the environmental risk factor, which is the infection with the murine leukemia virus, and endogenous retroviruses, and it's a multi-hit model. You have to have all 3 hits to get the disease. So it's quite an intriguing, naturally occurring model. So if we go back to this, that should say 1, 2, 3, 4, 5, 6 along the bottom where it says steps. You can imagine you've got the gene time environment model, but along the bottom, you've got the 6 steps. Okay, that should say 1, 2, 3, 4, 5, 6 in that blue bit down here. And you can imagine some people are born with lots of little risk factors, and some people are born with one very big genetic risk factor that takes them a long way, and then they don't need much else to trigger the disease. But what that tells you is, if you really want to find environmental risk factors, you shouldn't be looking in the people without a genetic cause or without an obvious genetic cause, because they have 6 steps to get. And we already know lots of different environmental factors can cause the same molecular step. You'll be looking for ages. But if you look in the people with a big genetic cause, they maybe only need 1 or 2 environmental risk factors or molecular steps to take them on. So they would be the people you should really look for the environmental risk factors on if this model is correct. So just to finish off, genetic and environmental risk factors we know contribute to ALS risk. That's possibly through a multi-step process. The themes of ALS cause appear to be DNA and transcriptome damage, cholesterol metabolism, neuroinflammation, and then novel antigens. So the exposure of these retroviruses or the cryptic exons may be what's triggering the neuroinflammation, for example. So these are the people who've helped sponsor my research. And more importantly, this is the team that did all of the work that you saw. Thank you very much.
Thank you, sir. No, there are no questions for the oration. So please be seated. Thank you for the wonderful oration, sir. May I request all our distinguished chief guests, our beloved director, Professor Prabhachandra ma'am, Professor Satish Chandra sir, Professor Nalini madam, and to please come on to the stage. Thank you, sir, for that excellent oration. So I'm sure that all of us researchers here, all of us from different specialities and various fields have learned a lot. Sir has taken us on a journey of motor neuron disease, starting from, may I request Professor Madhunath sir also to please come on to the stage. Yeah. So I'm sure all of us here from different fields have learned quite a lot. Sir has taken us a journey, starting from the clinical aspects to the genetics and the evolving concepts. And it was wonderful listening to the research which he has done, the genome-wide association studies, the exome-wide association studies, the epigenome associations. And it was truly a pleasure listening to you, sir. We were really honored to have you here. And also, as the title showed, the mystery of ALS. He showed us the real mysteries, genes, time, environment. We all know about the heterogeneity of the ALS, the complexities. And I was fortunate to listen to sir on 2 occasions before in Pactals. And one of my favorites is, of course, the multi-step hypothesis. And it's quite interesting how sometimes the 6-step hypothesis goes to 2-step, and how sometimes a big gene can lead to a longer time environment, shorter time environment, or how other multiple genes can lead to vice versa. So it was quite a pleasure listening to you, sir. And also, he showed us how these mysteries can be solved, or are being tried to be solved, how the therapeutic landscape is evolving, and how various therapeutic targets are aiming at the neuroinflammation or at various pathophysiological mechanisms. And we have a great hope for the future in the management of these patients. And of course, one more word about his slides, sir always makes it a point to put some picture of the environment or something which carries a deeper meaning. Possibly that retrovirus slide, I hope, was showing something how maybe suddenly one of it gets expressed. So there are deeper meanings to his slides too. Thank you, sir, for that excellent oration. We all have learned a lot. Let us give a huge round of applause for that excellent oration. I request all of you to stand up and please give a round of applause for a wonderful orator. We are truly privileged to have you here, sir. So now it is time for us to felicitate the orator. I request our director, ma'am, Professor Prabhachandra, ma'am, Satish Chandra, sir, our head of the Department of Neurology, respected Professor Madhunath, sir, Nalini, madam, to please come onto the stage and to felicitate him. Thank you all. Now may I request Professor Gourie-Devi ma'am, who has joined us online, to please speak a few words.
Thank you, Professor, for your excellent oration, fascinating work over the many decades of research. What you told us is the interplay of various factors, genetics, environment, multi-step, retroviruses, even cholesterol and all that, and neuroinflammation recently. So now we hope to find new targets for treatment, which you've already outlined about the neuroinflammation. And as we see in India, somehow we feel that the number of cases of ALS are on the increase. We have not done a population-based epidemiological study, but in the hospital practice, over the years, I seem to be seeing many more, or maybe there is a greater awareness among the physicians referring or the neurologists. So I think it's a great concern to us. And in the absence of any definite drugs to either reduce the progression, to slow down the progression or reverse, a lot of palliative care is becoming increasingly important. And particularly in India, with the socio-economic status, many of the palliative measures they cannot be taken care of unless the society itself helps. So I think that is where the challenges are. And one small fact, of course, we have not done very wide genetic studies, but the familial forms in the clinical practice, we have seen about less than about 2%, we would say. Whereas the figures in the Western and your own figures was something like 5 to 10% of familial. Is it really a true fact, or we need to do many more studies across the country? Thank you very much. You have enlightened us, focusing on multiple factors which may influence the disease progression, survival, and all that. Thank you. We look forward to many more years of productive work and hopefully to find a cure or something which can reverse the process. Thank you once again for a wonderful oration.
Thank you, ma'am. So now let me invite Dr. Saraswati Nashi, Additional Professor in the Department of Neurology, to propose the vote of thanks.
Thank you, ma'am. On behalf of the Department of Neurology, it is my privilege to propose the vote of thanks at the conclusion of the third Dr. M. Gourie-Devi Oration. We are deeply honored and grateful to Professor Amar Al-Chalabi for this enlightening talk. And I think you have taken us through ALS for the last 2 days. And it's exceptional work. It's a dream for me to work in a atmosphere created by you. So I think that is, so interaction with sir was really inspiring for all of us who were with him. I extend my sincere gratitude to Prabha Chandra Madam for her continued leadership in fostering academic excellence in NIMHANS. My also thanks to Dr. Nalini Madam for the gracious introduction of our orator and her persistent efforts in the care of ALS patients and ALS research in India. So it's a special privilege for all of us to celebrate this oration in honor of Dr. Gourie-Devi Madam. And as Sir said, Madam has continued to inspire generations of neurologists. So we are delighted, Madam, by your presence here, and we are grateful for the enduring legacy you continue to build. I would like to thank all our faculty members, our residents, our distinguished guests today who are online also, and also our faculty from other departments who are collaborating with us in research, from the Department of Neurophysiology, Department of Human Genetics, and everybody else in NIMHANS. My special appreciation goes to the organizing committee, the administrative staff, the convention center personnel, the audiovisual team, and the photography section, and everybody else who helped in organizing this meeting. With these words, I once again express our heartfelt gratitude to Professor Amar Al-Chalabi, Dr. Gourie-Devi Madam, and everyone who contributed in making this event a memorable and intellectually stimulating one. Thank you, and please join us for the high tea now.